First of all, I will talk about the history of UVB spectrophotometer. It is developed in the 1930s to study vitamin, especially in the vitamin A. First commercial UVB spectrophotometer is the Beckham DU, which launched in 1941. The use of this device is to identify and quantification of organic and inorganic compound. And it is also monitoring chemical reaction and kinetics. And also determine the purity and the concentration of the solution. The last one is to study photochemical processes. The principle of UV spectrophotometer have 5. The first one is observation of light. Second one is monochromatic light. The third one is Beer-Lambert law. And the fourth one is detector. And the fifth one is quantitative analysis. Next, I will talk about the working principle of UV spectrophotometer. The first one is light source, which is a high-intensity light source that emits light in a wide range of UV and visible wavelength, typically xenon lamp or a combination of deuterium and halogen or tungsten lamp. The second one is wavelength selection, which is a monochromator separate the broadband radiation from the light source into a narrow band of wavelength that can be used to analyze the sample. It is contain a prism or grating that disperses the light and allow only a specific wavelength to pass through the exit slit. And the third one is sample analysis, which is the monochromatic light passed through the sample, which may observe certain wavelength depending on its composition and its concentration. The amount of the light absorbed is proportional to the concentration of the observing species, as described by the Beer-Lambert law. And the last one is detection. After passing through the sample, the light intensity is measured by the detector, typically a photomultiplier tube, PMT, or photodiode array. The detector converts the light into an electronic signal that can be processed and displayed in an absorption spectrum.
UV visible spectrophotometer have 5 main components, which is light source, monochromator, sample holder, detector, and signal processing. The light source emits broad spectrum of UV invisible light, and it could be from tungsten, halogen lamp, or UV light. The monochromator disperses light passing through into its component wavelength. It consists of prism to select specific wavelength of light. In the sample holder, the selected wavelength of light directed through the sample. The sample absorbs light depend on the concentration of absorbing species in the sample. The detector measures the light passes through the sample. It converts the light signal into electrical signal. An example of detector is photodiode and photomultiplier tubes. Finally, the signal processing. It processes and displays the electrical signal as absorbance value, which is directly related to the concentration of absorbing species. Next, the procedure of sample measurement using UV spectrophotometer. Firstly, the blank. This blank is used to measure the intensity of light transmitted through solvent. Firstly, choose solvent, for example water, as a blank and add into cuvette. Secondly, wipe the cuvette and ensure no bubbles and insert the sample into the sample holder. Then, set the spectrophotometer to desired wavelength and record the absorbance. Then, adjust the instrument to read 0. Finally, check the blank to ensure the baseline remains at 0 during the measurement process. Next, the sample preparation. Firstly, ensure the sample solution is well mixed and free of bubbles. Then, pour the sample solution into another clean and dry cuvette. Then, ensure the cuvette is filled to the appropriate level and no bubbles or particulate matter. Lastly, the sample measurement. First, remove the blank cuvette and replace with the cuvette sample. Then, ensure the clear sides are aligned with the light path. Finally, close the sample compartment lid and record the absorbance value of sample at the selected wavelength. So these are how the UV spectrophotometer detects signal. Firstly, the light from the source is directed into the monochromator. Then, the monochromator separates the light into its component wavelengths. Then, the selected wavelength passes through the sample where part of the light is absorbed. The remaining light after passing through the sample is then detected by the detector. The detector converts the light into an electrical signal. This signal is then amplified and processed to give readable input in form of absorbance or transmittance. So this is how UV spectrophotometer works. A lamp provides a source of light, could be from tungsten halogen lamp or UV light, and the beam of the light strikes the diffraction grating, which works like a prism and separates the light into the component wavelength. The grating will rotate, so only a specific wavelength of light reach the exit slit. Then, the light pass through the samples. The detector will then measure the transmittance and absorbance of the sample. The transmittance is the amount of light that passes completely through the sample and strikes the detector. The absorbance is the measurement of light that is absorbed by the sample. The detector, sen- the detector senses the light transmitted through the sample and convert the information into a digital display.
In a UV-Vis spectrophotometer, results are determined by measuring the absorbance of light at specific wavelengths as it passes through a sample. After the intensity of transmitted light is measured compared to a reference, the absorbance A is calculated using the formula A equals to minus a negative log I divided by I naught, where I is the transmitted light intensity and I naught is the incident light intensity. For the data analysis, for spectrum, a plot of absorbance versus wavelength is generated. After that, peak identification, where peaks correspond to specific electronic transitions. And lastly, quantification, using the Beer-Lambert's law. Concentrations of compounds are determined from absorbance values. Beer-Lambert's law, also known as Beer's law, is a fundamental principle in spectroscopy that relates the absorption of light to the properties of the material through which the light is traveling. The law is typically expressed in the following form, which is A is the absorbance, Epsilon is the molar absorptivity of or molar extinction coefficient, C is the concentration of the absorbing species in the solutions, and lastly, L is the path length of the sample cell.
For the discussion part, I will discuss about the UVVs and nanoparticles relationship. So, nanotechnology has no direct impact on the working principle of a UVV spectrometer. However, it plays an important role in improving the analysis of nanomaterials employing UVV spectroscopy. So, for the nanoparticles characterization in UVVs, it will determine the size and shape. When nanoparticles exhibit surface plasmon resonant SPR, this creates a peak in the UV spectrum. The position and width of the peak are highly sensitive to the size and shape of nanoparticles. For the concentration analysis, the intensity of light absorbed by the sample is directly proportional to the concentration of nanoparticle presence, allow for quantifying the amount of nanoparticle in a solution. So for aggregation detection, when nanoparticles clump together, their interaction with light changes. This can be observed as a shift in the peak position or a broadening of the peak in the UV spectrum. And for the last part, background measurement, where the semiconducting nanoparticles, the UV spectrum can reveal the band gap energy. This energy difference between the valence and conduction bands, which determines properties like conductivity and light absorption. As for the application of UVV spectrometer, UVVs will be used in biomedical application for DNA and RNA analysis, also protein analysis. This UVV spectroscopy is used to quantify DNA or RNA concentration, monitor their purity, and even check for structural damage. Next is material science, where it will be used in semiconductor characterization and polymer analysis, where it will help identify different types of polymers and monitor their degradation process. For the environmental monitoring, UVVs will help in water quality analysis and soil analysis, where it can assess the soil organic matter content and identify potential contaminants. Also can detect pollutants like organic compound, heavy metal, and certain types of microorganisms by analyzing their light absorption patterns. And for the last application, it will be used in food and beverage industry for quality control and roughness determination, where the UVVs will be used to ensure product consistency, detect impurities, and monitor color and flavor properties of various food and beverage products. Also, changes in fruit and vegetable color during ripening can be monitored using UVV spectroscopy. As for pros of using UVV spectroscopy, it is simple and inexpensive, where compared to other instrument, it's inexpensive compared to other instrument. Also, it's simple, which doesn't need complex technical requirement. Next one is non-destructive analysis, and the last one is rapid analysis, which can save time. So for the cons of this instrument is sensitive to changes in temperature and pressure, can be only used to analyze substances that absorb light in the ultraviolet or visible region. And the last one, it can only be used to measure a solution, not gas or solids.
So the last part is the conclusion, which is the UV spectrophotometer measure light absorbing a substance by detecting light absorption at the specific wavelength, which is following the Bird-Lemon law. This technique is simple, cost-effective, and the best for homogeneous sample requiring careful calibration. Despite the limitation, it provides quick quantitative result, making it valuable in a many scientific fields.